Hey there, welcome back. In our last few videos, we have focused on the specifics of DNA, such as replication, transcription, translation, and mutations. Today, we're going to talk about what happens with DNA during mitosis and meiosis. Mitosis is the process that a somatic cell goes through during cell division. Somatic cells are the name for all the cells in your body that are not reproductive cells. This is an important distinction because reproductive cells have their own form of cell division called meiosis. We will go over mitosis first, as it is a good segue into meiosis. However, before we can delve into mitosis, we need to quickly go over the cell cycle. The cell cycle is the stages of cell development that need to occur for mitosis to happen. The cell cycle can be divided into two main sections, interphase and mitotic phase, or M phase. Interphase is the longest phase and is made up of three stages called G1, S, and G2. G1 is our first growth phase and is where the majority of cell growth occurs. During this stage, the cell will become physically larger and will also grow extra organelles and molecular binding blocks that we learned about previously, such as our nucleotides, amino acids, and lipids. All of this growth will prepare the cell to sustain mitosis. Next, we have the S phase. This is where we see DNA replication, transcription, and translation. Our last stage of interphase is G2. This is our second growth phase and is notably shorter than G1 and S phases. G2 essentially puts the final touches in place for mitosis. After G2, we are out of interphase and into the mitotic phase, which we will be going into depth about. Before moving on to mitosis, there is another phase that you might hear about called G0. G0 is a rest phase, where there's no growth, no synthesis, or division. Cells will enter G0 phase if any mistakes are made in G1, and they will stay there until these mistakes are corrected. As a quick reminder, the whole process we just discussed is a cycle, meaning that after mitosis, we go back into interphase and so forth. With that out of the way, let's move on to mitosis. As we know, mitosis is the process that a somatic cell goes through during cell division, and it results in two identical daughter cells. To do this, there are many stages it has to go through. Let's first take a look at the state of the cell after our G2 phase. As you can see, we have our nucleus, as well as the nuclear membrane that encloses the free-floating DNA, which has been replicated from the S stage. Due to this replication, we now have pairs of identical DNA connected to each other, which we will call sister chromatids. Now, before we move on, we have to go over some terminology because this can start to get confusing. When a strand of DNA gets replicated during the S phase, we know that it creates an identical strand that stays connected. These strands will condense during both mitosis and meiosis to form this X shape that you may be familiar with. This X can be described as being made up of two sister chromatids, because once again, the two strands are identical from replication. The two sister chromatids are joined at what we call the centromere, which will come up later when discussing mitosis. If we wanted to refer to the X as a whole, we would call it a chromosome. During mitosis, there will be a point in which the chromosomes are split in half and the sister chromatids are pulled apart from each other. These will eventually become parts of different cells. When this occurs, each of the now single chromatids would be referred to as chromosomes. In summary, when the sister chromatids are connected, we refer to them as one chromosome. When they are pulled apart and inside of the newly divided cells, the separated chromatids are now each considered chromosomes. Still with me? Because there are a few more terms that we have to go over. Let's start with the term homologous. This is an image of the beginning phase of mitosis. Notice that there are 4 chromosomes. As you can see, there's a big blue chromosome, a big red chromosome, a small blue chromosome, and a small red chromosome. We will say that the blue chromosomes are from the paternal parent, and the red are from the maternal parent. The term homologous means very similar or same in structure, such as size and length. Therefore, when we have 2 chromosomes that are very similar or the same in size and length, such as the big blue and big red chromosomes, or the small blue and the small red chromosomes, we would call them a homologous pair of chromosomes. So, we could say that the cell has 4 total chromosomes, or 2 homologous pairs of chromosomes, each made up of 1 chromosome from the maternal parent and 1 from the paternal parent. The last terminology we have to go over is diploid versus haploid. First thing you have to know is mitosis begins and ends with diploid cells. The term diploid is used when you have 2 complete sets of chromosomes, which we just learned are called homologous pairs. The diploid number is represented as 2n. To best describe what I'm talking about, take a look at this image. Look familiar? This is the breakdown of chromosomes in the typical human diploid cell, also called the human karyotype. As you can see, the homologous chromosome pairs are labeled 1 through 22, with the 23rd off to the side, which determines the biological sex of the human. To find the diploid number, we take the number of pairs, which is 23, and multiply by 2 to get 46. If you chose to refer to the 23 pairs of chromosomes, that would be considered the haploid number, which is half of the diploid number. Although we will go into haploid later during meiosis, for now you should know that meiosis starts as a diploid cell and ends as haploid cells. This means that there are no homologous pairs of chromosomes in meiosis. The first stage is called prophase. Now, prophase is kind of interesting because there are two parts to it, an early stage and a late stage. Sometimes you will hear people combine the stages, whereas others might keep them separate and call the late stage of prophase prometaphase. In this video, I will refer to the two stages separately because in my opinion, I would have a lot of stuff to squeeze in if I combine them. Anyway, let's start with early prophase. In early prophase, the nucleolus disappears, the chromosomes start to condense, and we begin to see two pairs of homologous chromosomes. In this example, I will be using this number of chromosomes purely because it is easy to follow, not because this is how many chromosomes there are in the body. While this is happening, microtubules that are commonly described as the mitotic spindle begin to form. The mitotic spindle will be crucial for separating the chromosomes later in mitosis. In late prophase, or prometaphase, the nuclear envelope breaks down and the chromosomes become even more compact. The mitotic spindle further develops to the point where it is long enough to grab onto some of the chromosomes. The microtubules are able to attach to the chromosomes by a protein called the kinetochore that lies on both sides of the centromere. The next stage is metaphase, or as I like to call it, middle phase. In this phase, the rest of the chromosomes become attached to the microtubules on both sides and are lined up in the middle of the cell. The middle section is called the metaphase plate. Now, when I was taught this, I was under the impression that the metaphase plate was a physical landmark, but it's just a term and there's no actual line. As long as all the chromosomes are secured in line, they are now ready to divide. This brings us to the next phase called anaphase. In anaphase, the chromosomes are split in half into two sister chromatids and pulled to either side of the poles by the attached microtubules. Now, as you can see, there are a few microtubules on either side that are not attached to chromosomes. These push against each other, distancing the poles and elongating the cell. Next, we have telophase. In this phase, the mitotic spindle breaks down and we begin to see the structures of a nucleus forming. This includes the formation of the nucleolus and the nuclear envelope. We also see chromatids start to loosen up. Lastly, we have cytokinesis. Cytokinesis literally translates as the movement of cells. This stage actually starts around anaphase when the cells were being pushed away from each other by the microtubules. Cytokinesis is completed once the cells split into two. The area in which it begins to split is commonly referred to as the cleavage furrow and begins to form in telophase. After the cleavage furrow completely closes off, we're left with two identical daughter cells. These cells are now ready to go through the cell cycle once again. Thankfully, if mitosis made some sense, then meiosis shouldn't be too difficult to understand. Although there are some notable differences you should be aware of. Meiosis is the cell division process that reproductive cells go through to create sperm and egg cells with the intention for them to undergo fertilization later down the line. As you know, our DNA is a combination of both of our biological parents' DNA. For this reason, the end product of meiosis results in 4 genetically unique cells with one set of unpaired chromosomes. Remember when I brought up haploid cells earlier? I just described exactly what one is. Haploid cells have one unpaired chromosome, which you will often see represented as N. These unpaired chromosomes have the potential to combine with other haploid cells during fertilization and create a diploid cell, which develops into an offspring. Now, one of the biggest differences between mitosis and meiosis is that meiosis occurs in 2 cycles. These cycles are referred to as meiosis 1 and meiosis 2. The reason for this is that we can obtain genetically unique cells rather than identical ones seen in mitosis. After all, we all want to be unique individuals, don't we? If meiosis didn't have 2 cycles, we wouldn't have nearly as much variation as we currently do. Not to mention, it really helps with the fitness of an organism, but you don't need to learn that yet. Similarly to mitosis, meiosis 1 begins with prophase 1. Also like mitosis, we start with the diploid cell, or 2N, which means that we have two sets of homologous chromosomes. In prophase 1, the two sets of homologous chromosomes pair off. Once they pair off, they get very close together, which allows for the chromosomes to swap a piece of their DNA. This process is called crossing over. Once the DNA fragments have crossed over, recombination of alleles occurs. This is the first mechanism in meiosis that increases variation. Keep in mind, while this is happening, we are also seeing the same changes in the cell that occurred in mitosis, such as the breaking down of the nuclear membrane and nucleolus, as well as the formation of the spindle, in which this case is called the meiotic spindle rather than mitotic spindle, for obvious reasons. Moving on to metaphase 1, in this phase, the homologous chromosome pairs line up side by side in the middle at the metaphase plate. The side in which the homologous chromosomes are lined up on is completely random. This further aids in creating variation. Next, we have anaphase 1. In this phase, the homologous pairs are pulled apart to opposite poles of the cell. Now we should have one big and one small chromosome on either side, both of which are made up of two non-identical sister chromatids from recombination in prophase 1. Next, we have telophase 1 and cytokinesis. This is where the meiotic spindle is gone and the cells split into two at the cleavage furrow. In mitosis, this would also be the part where the nucleus reforms. This can still occur in meiosis 1, however, oftentimes the nuclei will remain dissolved because they are about to undergo another round of meiosis. So after all of meiosis 1, we're left with two haploid cells, each having two non-homologous chromosomes. The two haploid cells will now each undergo meiosis 2, which is quite similar to mitosis. Meiosis 2 begins with prophase 2, in which the two non-homologous chromosomes condense. Then they will go through metaphase 2, where they once again line up at the metaphase plate. Next, they go through anaphase 2, where each pair of sister chromatids are separated and pulled to either side of the poles. Lastly, we have telophase 2 and cytokinesis, where the cells break off and the nuclei are reformed. As you can see, we are now left with four genetically unique haploid cells, each containing two chromosomes that are both made up of one chromatid. That just about sums up what you have to know regarding mitosis and meiosis. Hopefully this helped you understand the basics surrounding how our cells are able to duplicate themselves. Thanks for watching! In our next video, we will discuss genetic inheritance with a focus on Mendelian genetics and Punnett squares.